Good morning, good afternoon, and good evening, everyone. I'm Elizabeth Nolan. On behalf of Defined Health, we welcome you to today's edition of Defined Health Insight Series webinars. Today, Jeff Bachman, Mike Rice, and Joel Sandler, who lead the oncology practice of Defined Health, will lead a discussion entitled, Reconsidering Relevance in Oncology. Is the market immune to your non-IO assets? Defined Health is pleased to bring you our Insight Series webinars as a way of showcasing how we view and think, looking forward, about various critically relevant topics for the biopharmaceutical industry. Our viewpoint is the result of our 25-plus year history, consulting with hundreds of pharma, specialty pharma, biotech, and investor clients. In these engagements, we bring our unique perspective grown over several decades of evaluating pipeline and inline opportunities by bridging our knowledge of drug development, business development, and commercial strategy across multiple therapeutic categories and technology platforms. I want to quickly call your attention to two of our in-person events coming soon. Defined Health will present the 27th Annual Cancer Progress Conference in New York on March 8th and 9th. Please visit our website, cancerprogressbydh.com, to view the outstanding program, which includes world-class oncology luminaries from academia and all walks of industry, clinicians, pharma, biotech, diagnostics, big data, payer, regulators, advocacy groups, not-for-profit organizations, and investors. Panels will focus on immuno-oncology, melanoma, myeloma, N-of-1 trials, and RNA as a target and a therapeutic. Please register at cancerprogressbydh.com. Therapeutic Insight by Defined Health will be held at EBD Group's BioEurope Spring Partnering Conference that will take place April 4th to 6th in Stockholm. Our sessions will cover drug pricing, neurodegeneration, inflammation and fibrosis, and novel therapies in diabetes. We hope to see you at our sessions or in the partnering booth. To learn more about Defined Health, our capabilities, and our upcoming annual Cancer Progress and Therapeutic Insight conferences, please visit our website at www.definedhealth.com or send us an email or give us a call, and we'll be happy to tell you more. Now for some housekeeping. After this discussion, in a couple of days, audiovisual and PDF versions of the program will be available on our website. We should have time for questions at the end of the presentation and discussion. Please send them along as you think of them, and if we don't get to them in this hour or so, we will follow up with you in the next day. Thank you, and enjoy this Insight Series webinar. Joel?
Great, thanks, Elizabeth. So it's really a pleasure to be here and to discuss with you what we think is a very interesting topic and in somewhat of a unique forum, which I'll get into in a moment. Just to introduce the title, Reconsidering Relevance in Oncology, is the market immune to your non-IO assets? Just to kind of spare you the suspense, the answer, of course, is no. But it is somewhat of a provocative and hopefully not a doom and gloom title that drove anyone away. But as you'll see, it's somewhat of a contrarian question these days, and not contrarian for the sake of being contrarian, as we'll get into, as there are many of us, or many of you out there with portfolio assets that are not classically considered IO assets. And the question really ultimately is about how one should be thinking about positioning these and what are the different strategies that have been taken to date. You know, how are things evolving and where are things moving in the future. And so as Elizabeth mentioned, this is all kind of a run-up to our very exciting Cancer Progress meeting that's taking place in about 3 weeks from now in New York. And we hope to see many of you there. To the extent that those of you who cannot attend, hopefully at least this hour will provide you with a sense of the type of insight that Defined Health can provide when weighing in on some of these more strategic, high-level, and also nitty-gritty questions And today, of course, speaking within the context of oncology, of course, we touch on many other therapeutic areas in our work as well. But with regards to CP and, just kind of tying that into our discussion today, I think it's important to point out that we will be touching on many of the themes that will be expounded upon in 3 weeks' time, including touching on different therapeutic areas, novel, unique mechanisms of action, whether they be IO or non-IO, and we'll touch a little bit on RNAi, and ultimately, the big question of value, kind of the big elephant in the room, so to speak, and of course, we'll have a big panel on that in a couple of weeks. So we hope to see you there as well. So just to introduce the players, so it's myself, and I'm Joel Sandler. I'm pictured on the right there with who would be playing me in the Hollywood version of this story, David Schwimmer. We also have Mike Rice, who unfortunately cannot be played by his doppelganger, who unfortunately passed away a few years ago. And Jeff, I guess Spielberg will be directing our movie.
Thank you, Joel.
This really is a unique format for us. It's 3 consultants speaking to some of these different issues. I hope to be somewhat provocative. We can discuss, we can debate, we can disagree, and really, in doing so, hopefully get to the heart of some of the questions that many of you are thinking about with regards to your non-IO assets in the context of what's rapidly become an IO universe. So this slide really needs no introduction. As many of you know, immuno-oncology has arrived, and it's certainly what has many people, many different stakeholders, both in the investment world, the partnering world, clinical world, maybe not the payer world, although we can speak to that. Although certainly they're excited about the prospect of cures, but the price tag that's going to be attached to that, I think is going to be an interesting topic of discussion in years to come. But certainly IO is a very interesting area, very groundbreaking when thinking about some of the different modalities that are emerging, like checkpoint inhibitors, CAR T-cell, adoptive cell transfer technologies, and the like. And some of the dramatic results that are coming out in the clinic with these different modalities. Just to throw a little bit of cold water on that notion, though, and the idea in general that has everyone excited these days. The idea about or the excitement around potential cures in cancer is nothing new. It kind of goes back at least 100 years with things like radium, different surgical techniques, of course with the advent of chemotherapy, combination chemotherapy, and people like Mary Lasker and Sidney Farber, maybe three or so decades ago, talking about curing patients in what were previously considered incurable tumor types, cancer types. And now, of course, with IO, a lot of excitement again, and the broadening of the KM curves and the like. And so there is this glimmer of hope that we might be able to cure the broad majority of patients. But I do want to point out that people do tend to get very excited about these things very quickly. And of course, while the data is very striking and very exciting, and I'm showing here on the left some data that was presented recently by Jed Walchuk at Memorial Sloan Kettering, showing combination of ipilimumab versus ipi-nivo, so a dual checkpoint combination in advanced metastatic melanoma, where you really do see these dramatic and unprecedented effects. As well as the very high-profile cure, or at least durable remission achieved by the very elderly, but of course very eminently lovable Jimmy Carter as a result of treatment with pembrolizumab. It's certainly very, very exciting. At the same time, we're on a collision course, or maybe we're already there, with some people who are becoming increasingly vocal about concerns around sustainability, and if one can argue that we've made and priced at a premium the incremental advances in oncology over the years, and now we're faced with a situation where we're curing patients, and who's going to pay for all of this? I think that one has to take stock and be aware of the fact that anyone who's developing cancer drugs, has to think about it in this context. With regards to IO itself, in parentheses, I say modern IO, and of course Coley's toxins, and the idea or the correlation between acute infection and cancer cures has been around for a long time, since the 19th century, if you can believe that. But the modern advent of IO is really just in the last 20 years, and the frenzy really in the last, I would say, 2 or 3 years. And so, I think that overnight, both perceptually and in reality, things have changed. And it's important to understand how those with non-IO assets are reacting to those changes. And of course, we're not talking about black and white. Many companies have both IO and non-IO assets, and many of the so-called non-IO assets are influencing the immune response, if not all of them. And so I think that's an important theme that we'll want to touch upon today as well. And so, getting ahead of myself, so maybe many of these non-IO assets are in fact on the IO spectrum, to borrow a term from other therapeutic areas, where if you think about the fundamental goals with immuno-oncology and immunotherapy in cancer, this analogy of stepping on the gas, which is depicted on the left, and then taking your foot off the brake, where you have tumor-infiltrating lymphocytes that are not really doing their job because of the cancer cloaking mechanism or within the tumor microenvironment. The objective is to kind of hit on both of those aspects of the immune response in order to achieve a more durable anti-cancer effect, hopefully one that's within a viable therapeutic window. And obviously there are a number of IO agents that have emerged, ones that I've already mentioned, that have really kind of exemplified this ability. Vaccines have been around for quite a long time, as well as cytokines and their ability to rev up the immune response. But it's really with the advent of the checkpoint inhibitors and their ability to kind of take the foot off the brake with regards to the checkpoints that are present either at the immune synapse or in the lymph nodes, and in order to really unlock the full potential of immunotherapy. But there are other assets and classes of compounds that have been around for quite a while that are already doing this, maybe not in isolation, but in combination with some of these newer IO agents. And so, I think this all kind of enters into this discussion of how one takes their non-IO assets, their VEGFs, their TKIs, their reformulated chemotherapy agents, and thinks about how to move forward, short of just kind of putting them on the shelf and walking away from non-IO en masse. I think it's really important to think about the need and the potential for these two different broad buckets to complement one another. And so to kind of drill down on that point a little bit further, when you kind of break down, and again, these slides will be made available afterwards, so don't feel the need to scribble furiously, but if you think about some of the different aspects that are important to anti-cancer therapy with regards to patient selection, efficacy, safety, combinability, each of these different broad asset classes bring something to the table. And I think it's important to point that out. So with regards to patient selection, targeted therapy really introduced the idea of patient selection to begin with. And the hope that one could administer precision medicine with the use of companion diagnostics to select patients better or predict which patients are going to respond to a particular therapy. In the case of IO, for example, with checkpoints, it's a little bit more challenging, or at least at present, to predict which patients are going to respond. The immune system is more plastic than genetic perturbations, and therefore there's the potential to amp up the immune system and increase the expression of those biomarkers that are going to allow for an effective anti-cancer response. And of course, there's emerging evidence that mutational burden, neoepitopes, really do drive the anti-cancer effect of IO. With regard to efficacy, of course, when compared to the tailing effect on the Kaplan-Meier curves with the checkpoint inhibitors, some of the targeted therapies people have perceptual biases with regards to transient response and resistance, and one asks whether it's possible to address tumor cancer heterogeneity with a targeted agent. But let's not forget that some of these more aggressive approaches like chemotherapy may be required to debulk the disease in combination with resection, and that then sets up a success story with IO. With regards to tox, there are non-overlapping toxicities. A lot of the immune-related adverse events have been kind of downplayed, and not surprisingly, because you, to some extent, rely on that tox in order to achieve an anti-tumor immune response. So it's difficult to kind of parse those out. In contrast to that, non-IO, obviously chemo has kind of a classic suite of adverse events that impact quality of life. Targeted therapy and monoclonal antibodies are relatively benign, and so as we'll get into, the ability of these to be layered onto IO and the need for complementarity between the two, having that benign or relatively benign safety profile makes this far more viable than it would otherwise be. And then finally, with regards to combinability, you take multiple targeted therapies or chemo agents in a classic chemo cocktail to control resistance or drug resistance to the therapeutic regimen. Whereas in the case of IO, the complementarity between a vaccine and a checkpoint, for example, is thought to be required. But maybe complementarity between some of these non-IO agents like TKIs and chemo to achieve an in situ antigen release that can then be leveraged by the IO agents to really achieve that durable response. So I said at the outset that this is a contrarian discussion, but it's not contrarian for the sake of being contrarian. And so there are a lot of non-IO assets out there, and we can get into the question of how we define IO versus non-IO. But a quick poll from the databases that we rely upon illustrates the fact that yes, the majority of growth is going to be driven by IO, but the majority of revenues, even out to 2020, is still coming from non-IO, and the majority of pipeline assets is still non-IO. So if you're in the position where you're thinking about how best to develop your TKI, how best to position your platform technology that isn't classically IO and maybe is going after some quote-unquote undruggable anti-cancer targets. What are the options out there for you? What have people done? And what are kind of our thoughts in terms of how one can best position themselves? So those are some of the questions I hope to get into today. And here, just I think kind of the bottom half of the slide kind of speaks for itself. We talk a lot with regards to the second bullet. The third bullet on the left. If one wants to kind of acquire their own suite of IO agents and checkpoint inhibitors and the like, obviously that's very appealing from a pricing, market access perspective. You can kind of set the price of your combination regimen, but at the same time that requires a lot of upfront capital. So there's a bit of a trade-off there with regards to non-IO. Or you kind of relegated to these unique niches where IO, it seems to be high-hanging fruit for immuno-oncology, or is there are there other options out there and ways to kind of get past this perceptual bias about durability? So these are some of the questions I want to get into, and the format of our discussion really is to kind of set the stage and then walk through some different case studies, and then I'm going to ask my panelists, Mike and Jeff, to really weigh in when we get to some of these different case studies. But I think these questions, and I've touched on a number of them already, so I'm not going to walk through them. I'll just emphasize, if there are any questions from the audience, please feel free to chime in, and we'll try to either answer them in real time or after after the discussion, we'll get back to you. Okay, so and these are all pretty modern or pretty recent things that have come out in the press, and the hope is that they'll be illustrative, provocative, and serve as the basis for some discussion. I'm not going to walk through the slides in the interest of time. Again, you'll have these available to you. But to start, here's an example where 5' very publicly announced that they were taking their FGF trap, which had some clinical data, or has some completed trials both in non-small cell lung cancer and mesothelioma. You can argue that checkpoints have a presence in both of those, but certainly much more so in the case of non-small cell lung. And this asset, which is partnered with GSK, the partners publicly decided that they were going to stop their pursuit of non-small cell lung cancer because of what they see as preponderance of IO in the space currently and going forward, and really to kind of double down on mesothelioma. So, Jeff, I'll start with you. Do you think that this is, first of all, is it a strategy that a lot of people are taking, whether they're publicly admitting it or not? And if so, what are the implications for people who are in a similar situation?
Thank you, Joel. Thank you for that intro. I don't know that I could make any sweeping statement about what people are actually doing or maybe have reported on, but I would say that it would be, I think, pretty clear to me that anyone in this space would need to be strategizing around the potential impact of IO on their programs, whether they are non-IO or on IO for that matter. And in fact, this is kind of what we do day in, day out here at Defined Health, or part of what we do when helping clients think about, the kind of clinical development path they could pursue, or trying to identify potential white spaces or way to combine agents. Is, we need to try and read the tea leaves on how future management of these diseases are evolving, and that's not just for its potential impact on the potential utility of a product like this, the 1039, but also on its actual clinical development. And there certainly have been many examples, not in oncology, only, where one does have to kind of take a step back and say, well, the standard of care has suddenly changed, and we need to think really hard about how we're going to move these programs forward. So I think it was very interesting that they did this stance. One from the outside might have thought, and I haven't done the search around kind of the scientific basis for what I'm going to say, but I'm going to assume that GSK and 5' did. To what degree there might have been some actual synergy between targeting FGF and combinations with IO agents, just like now it is evolving of the potential role of anti-angiogenic agents, or certainly bevacizumab and others. So I think this is something that we're certainly going to be seeing more of, and I think it's well exemplified in particular with kind of the change in standard of care that happened in a relatively short order, and the kind of the rapid evolution of standard of care in a short period of time in melanoma, between the dramatic unmet need, the launch of the first and then subsequent BRAF mutation inhibitors and MEK inhibitors, and then the major change with both the launch of anti-CTLA-4 agent IPI, and then followed by the PD-1s, and now the combination where in those combinations you become almost the mutation status almost becomes irrelevant. I'll stop there and let the rest, Mike, and also you chime in, Joel.
Yeah, thanks, Joel. Jeff. This is Mike Rice. I just want to add to what Jeff was saying about there is some risk in skating for the immuno-oncology open ice to say for areas where it looks like immuno-oncology is not currently having a big impact. So I spend more time in the heme malignancy side of things. So the example that just brought to mind was multiple myeloma. As of ASCO last year, post-ASCO, there were big headlines that didn't look like checkpoint blockade was going to have a big impact in myeloma based on some of the initial studies as monotherapy. However, just several months later, by the time ASH rolled around, a combination of pembrolizumab and lenalidomide had response rates of near 80% in double refractory patients. So really a striking turnaround and impact for that. So in that period of time, it was interesting to hear some drug developers in the myeloma space talk popping corks that their non-IO asset was positioned for this potentially white space opportunity, but it's just that those white spaces can quickly close if you don't have a contingency of how you're going to deal with the changing IO landscape.
Yep. All right, good. I mean, I think FGF's an interesting one. I think about things targeting the tumor microenvironment, which FGF can arguably be placed in that bucket, and there is evidence to suggest that, for example, targeting VEGF and VEGF receptors, there's some synergy there between IO and non-IO, but I think 5' kind of made this decision whether or not that they'll end up sticking with mesothelioma and or perhaps going elsewhere. I just think it's an interesting exemplar. Let's move on to the next one in the interest of time. Okay. So, this was a really interesting one for me. When I heard that Briggs Morrison was leaving AstraZeneca to go to Syndax, and clearly with the intent of pushing forward their HDAC inhibitor, atinostat. To my knowledge, atinostat is, perhaps differentiated from other HDAC inhibitors, perhaps not. But I think there's always been discussion about how best one can develop the epigenetic agents, the hypomethylating agents, the HDAC inhibitors. And there's this suggestion that their value could be further unlocked in combination with IO. And so to me, this was a story of taking an asset class which has somewhat struggled, obviously with Feridac in multiple myeloma and Berinostat and some of the other HDAC inhibitors in the T-cell lymphomas. one can argue that it's been a bit of a slog, and here you have a company that's making a concerted push to say that this is an IO agent, or at least one that plays nicely with IO. They've tied up some really nice partnerships, and are kind of saying that no, HDAC inhibitors are in fact part of the IO landscape, or should be, and this is the example. So, Mike, I'll start with you. Are they retelling the story, or is this a story that's always been there and we just didn't know about it?
Yeah, so I think retelling the story is an important point of positioning your asset in the future. Obviously, it needs to be backed by the science, and it'll all pan out in clinical development anyway. But certainly, as far as gaining financing and moving forward, it's been a very quite successful strategy that some of the companies have taken. The role of epigenetics has long been known to be in tumor evasion and escape from immune surveillance and things like that. So obviously, mechanistically, it makes sense, and it was here. So I'm not sure. In this case, it may be a real winner that they've repositioned it and showing synergy with checkpoint blockade. But I'll pass to Jeff and Joel and whoever wants to comment next on it.
Jeff, what do you think?
Well, I mean, I think that not to simply stick to the story around the HDAC inhibitors.
Sure.
We have a lot of debate. So what really originated this webinar were many internal discussions about either what we saw from the outside as we looked at companies thinking about what appeared to be non-IO assets, put that in quotes, or the direct questions we were being asked by companies with those seemingly non-IO assets. And we would often talk about, and I've heard many others in the industry talk about, to the degree to which many companies are contorting themselves to represent their assets as IO. And I think it goes back to one of the earlier slides that Joel showed, that look, at the end of the day, it may be that almost everything, if not everything, somehow owes some part, varying degrees, in some cases maybe major part of their efficacy or activity to the involvement of the immune system, which, as we well know, would have never been really on the radar screen, not only because people weren't, kind of looking at that way, they had blinders on, but just the models themselves, the way these were studied, were not designed to elucidate or bring those elements of their mechanisms specifically to light. Now, you can take this to an absurd level and say, well, ultimately everything, if you follow, downstream far enough. But there are certainly many, many other examples that we can point to. I mean, Joel and I both alluded to the anti-angiogenic agents, and specifically anti-VEGF and modulation of the tumor microenvironment. But there's, many of these agents have been, that are already approved, and many of them are being kind of re-examined in light of their potential role, because many of these agents, antibodies and small molecules, do have effects on the immune system. Cetuximab can can affect and activate M2 macrophages on the one hand, or bortezomib can sensitize tumor cells to CTL lysis, so they're, one way or another, I think, we're going to be seeing more and more of these internal discussions, and you can even see it when you look at some of these biotechs where they've, kind of grabbed the reins very early on, meaning within the past several years, at the dawn of this, modern IO age, to kind of reposition and tell the story for many of their assets, which might be very well plumbed categories like epigenetics, or many of the intracellular pathways that TKIs that are either approved or still in development have been going after, to really promote them and do all the kind of preclinical studies to really demonstrate that, yeah, it's not just kind of incidental. These have some very important and significant immunomodulatory effects, and will be likely useful, in combination with established IO agents like checkpoint inhibitors.
Yep. No, I agree with that. And I think that there are certain classes that kind of lend themselves to that strategy more than others, many of which you've listed, where the potential is there, it's always been there, and it behooves the developer to kind of go back maybe to the preclinical models and demonstrate, in one or two experiments that, there is a role that, many have been kind of speaking to putatively or theoretically. We have the data to support it, and then to go out if they're looking to partner, at that stage to go out and kind of make the claim that we're in a position and we have the data to support it, and to get clinicians excited about patient accrual and combination studies, et cetera. So I think that's a very interesting strategy, and one that people need to be thinking about.
Let me just add one thing, too, if I may, Joel, before we go on, and that it's not, since we're talking about this idea of or this is part of this underlying theme of these agents, modalities, et cetera, that have an effect on the immune system, we want to be sure to emphasize, and you alluded to this before on a slide, that, many cytotoxic agents have not only kind of direct cytotoxic cell killing, but kind of beneficial effects to this end, like gemcitabine. And very importantly, radiation has very strong immunogenic cell death inducing activities, and a number of interesting studies around abscopal effects between combining RT plus various types of checkpoint or co-stims have been deployed. So, we're going to be seeing more and more of this, and I think it's very easy for some people on the outside to say, oh, these companies or whoever is just trying to kind of take advantage of this big wave around IO. But, many of these are very legitimate stories mechanistically and clinically in terms of their role in playing into enhancing or modulating to ultimately the benefit of an anti-cancer effect various immune agents.
Agreed. Okay. Let's move on. I'm going to quickly turn this over to Mike. My take on this deal, and I think this is a fascinating deal. Ibrutinib Pharmaceuticals early on, in 2011, I think signed a deal with Janssen to co-develop ibrutinib. That's a 50-50 split. AbbVie came in, bought out the company for, I think it was $21 billion 4 years later, and for 50% rights to the drug. So obviously a lot of value being attributed to that drug. Acerta comes in with a potentially best-in-class, but what struck me is that, AstraZeneca took them out. Clearly they're making a push to be one of the big players in the heme space. I think BTK inhibitors have potential in solid tumors, but to date, obviously, the story is all about the lymphomas. And so, a $4 billion buyout of Acerta. There aren't many other dedicated heme oncology players out there. One could argue maybe Bayer, but if you're trying to fill a gap in a franchise, and one could argue if you want to be a player in the heme space, that you should have a BTK inhibitor. And I don't want the discussion to be strictly around BTK, but are there opportunities with analogous assets out there or mechanism classes that one can kind of come in and fill that gap?
Yeah. So just a couple comments on Acerta and BTK. There's a couple things going on. So one is just that the BTK class, or just B-cell pathway inhibitors in general, are becoming, hopefully quickly becoming, foundational therapies in B-cell malignancies, mainly CLL, but also other indolent lymphomas and even potentially in aggressive lymphomas in other settings. So just one, it's becoming without an IO store, it's becoming a very significant drug class for B-cell malignancies broadly that is displacing the use of chemotherapy. So one, there's a very attractive avenue that as far as filling a gap. The other thing is just that if you're invested in checkpoint inhibitors in IO assets, and maybe having a lot of significant assets in the IO space, but maybe trailing other IO franchises in the space, having more combinations that you can bring to market yourself and to experiment with internally is going to give you a competitive edge to catch up and maybe get into other settings. So I think it was a very interesting deal. It's a very interesting asset. It looks promising. As far as other things out there, so there are other B-cell receptor pathway PI3 kinase inhibitors, other things that look interesting, that it could also kind of play in the same sort of way. I know a little bit less about the mechanistic synergies between these. Maybe you or Jeff could comment on that.
Yeah. Yeah, Jeff, is BTK, is that an IO target, or is that a kinase target? Is it a targeted therapy, or?
Well, from what I know, and again, I haven't researched it thoroughly, but from my understanding is, at least in terms of the effects that have been published on for ibrutinib, a lot of that benefit is believed to be due to the effects on T cells of ITK, which ibrutinib also hits. But I know that BTK inhibitor is important in T cells, obviously, and also, I believe, in dendritic cells. So, I think this this raises the issue that a situation where before, when everyone would have been studying these targeted agents for their direct effects on those pathways and as cytostatic or cytolytic agents, depending on their nature. And then we're talking about maybe just ones that are highly specific, but many of these do have other kinases that they do hit, and whether it's the main kinase they hit or sometimes the additional kinases they hit with lower activity, these do in fact have immunomodulatory effects, and so BTK is just one example, but certainly sorafenib, mTOR inhibitors, many of these others, as we were saying before, do have, when you study them for those immunomodulatory effects, you frequently find them.
Yeah, great. All right, moving on. So HER2, I have to say this is one that surprised me a little bit. I figured Roche kind of had the space locked up tight between Herceptin and Kadcyla, Perjeta. But, there's always been kind of this use of, there have been a lot of kind of anti-HER2 ADCs out there, maybe just to kind of prove the concept. I never really thought they were going to go anywhere commercially or even in terms of traction with partners. But more recently, there are things like the FC-optimized anti-HER2 mAb from from Macrogenics, this big deal that Bristol did with F-Star, and Mersana just announced a recent deal. So maybe there's still money being left on the table with HER2. Is that being driven by gaps in what Roche has done, or is it more kind of, when you're thinking about monoclonal antibodies, for example, and the potential for ADCC, and it's kind of being cast in a new light now, right, with the checkpoint inhibitors and everything else that's going on in IO. So Jeff, do you think that this is something that's going to open up doors where we thought they were shut?
Good question. Well, I think what we can do is first of all, I think the third bullet here is part of the key here, right? Because we all know that we're talking about, for high expression, only a portion of the breast cancer patients that are HER2 3+, and then in most published studies we're talking about even in those patients, when you use an anti-HER2 agent, you're still only talking about maybe at best having 40-50% response rate. So this unmet need that remains both for HER2 targeted patients that has not been fully addressed by the addition to that armamentarium of pertuzumab or Kadcyla, as well as the much larger population of non-HER2 3+ patients, suggests that there's a big area to operate in the space, whether it's through augmenting the activity of agents that target HER2 itself, or going after HER2 in different ways. So what we don't have on this slide is there's a lot of vaccine approaches that are and have been historically attempting to target HER2. And then, of course, there are other approaches to get at this that aren't strictly HER2 specific.
Yep. No, I agree with that. I mean, to be honest, I've certainly, maybe not in the last year, but in years prior on projects where I've had people saying, let's pursue HER2, I've always there's always a part of me that said it's not viable.
Yep.
Clearly, I can be wrong, and I think this is an area where Jeff, you and I have disagreed internally in the past. Mike, are there any aside from
I mean, I think the point is that there is no doubt that it is commercially crowded, and therefore ultimately the clinical challenge of trying to bring something forward that's going to demonstrate sufficient outcomes, not just incremental, to be able to fit into, possibly even supplant, whether it's extant agents or fit in alongside, following, or with them, because I think the issue still remains that for a sizable portion of these patients, they still will progress through lines of those agents and need something else. And we're just talking about the kind of classic HER2 plus 3+ population. So I think our kind of historical dialogue, let's call it, is as much around, well, what's it take to do that? Can a particular MOA or platform do it? Can a small biotech have the wherewithal to try and do that, or does it need a large partner? Will a large partner be willing to take on the Roche Genentech juggernaut, as BMS is certainly willing to do here? So I think it really revolves around that. It's not like it's an open and shut case.
Yep. I'm just going to move on in the interest of time, but Mike, I want you to speak to, and EGFR is obviously not unrelated from HER2. They're in the same family, and well, EGFR1 and HER2 are in the same family, of course. And one could argue that EGFR and lung cancer is one of the kind of remaining bastions of targeted therapy. At least at the moment, we have some next-gen EGFR inhibitors, AZ, of course, with Tagrisso. That's going to be a blockbuster, according to analysts. Clovis, unfortunately, had a setback, but they were kind of neck and neck with AZ. And I think part of this is driven by the fact that PD-1s, at least on the face of it, are less active in this subset of lung cancer patients. And so there's at least been a short to midterm opportunity for targeted therapy. So I don't want to necessarily get stuck on EGFR, but Mike, are there other examples, or maybe you want to speak to this one, where targeted therapy, in quotes, or other mechanisms of action, there's still some opportunities there, and opportunity might mean commercial, it might mean earlier than that. It might be perceptual, and therefore the ability to drive partnerships around a particular asset.
Yeah. So I mean, obviously, non-small cell lung cancer is a big high unmet need. Regardless of the targeted agents that are out there, patients progress rapidly and are seeking other things. And it really needs something disruptive like immunotherapies to come in there. So I think it's a great win for patients. But as these markets become more molecularly stratified and we individualize, personalized therapies more, I think there will be more. Both breast cancer that you just mentioned and lung cancer becomes more and more segmented, where we're now taking one cancer that was defined by its organ involvement or histology, and now breaking that into many subsegments, each with its unique potential therapeutic approach. So I think there are many programs that can be repositioned, or as we learn more about the genetic drivers of the disease that are going to kind of reshape the market and show some opportunities. And I think that's going to happen regardless of the impact that immuno-oncology is going to have. That may be another mode of treatment that will be sort of wrapped around whatever other individualized therapies will be offered to patients, such as the HER2 agents, the PARP inhibitors, and whatever, and breast cancer, and then the EGFR inhibitors and other approaches in non-small cell.
Yeah. And Jeff, I want to give you a chance to speak too, but I think the next example, and I paged on to the next slide just in the interest of time, further illustrates the point. So now you have PARP inhibitors, of which there are at least half a dozen in the clinic, all kind of presumably had been vying for a spot in triple-negative breast cancer, BRCA-positive, or ovarian cancer. Now you have this very kind of striking data coming out with olaparib in prostate cancer, coming out of authored by Johan de Bono, who's one of the most prominent KOLs in prostate cancer. Perhaps carving out for the first time the emergence of targeted therapy in prostate cancer. At the same time, we have this phenomenon that I alluded to earlier about mutational burden and it's the ability or the correlation with response to checkpoint inhibitors. So maybe this isn't a targeted therapy story, or maybe it's a targeted therapy story that gets augmented by checkpoint inhibitors. Jeff, what do you think about this?
Well, I think part of this is, so, there's lots of discussion, and, we don't have any direct points here, on kind of our list of issues in terms of the idea of biomarkers and stratification within IO, other than alluding to kind of the obvious place where we are right now. I was going to say where we are stuck. I'm not sure if that's the right terminology, but certainly, this whole PDL issue and relevance or not. And I think, what we will continue to see is the evolution of an understanding. We'll have new types of diagnostic types of tests that will evolve, whether from nanosystems or adaptive or other companies that will help guide, hopefully, decisions about when and how to use these agents, either upfront or through monitoring the course of treatment. But what I'm hoping is out of some of those tools will come additionally better understanding that will help elucidate the potential synergies or interactions, at least, between a number of these pathways and the IO space, like has come out, and making this link between mismatch repair deficiency, or ultimately maybe even just a broader view of HRD, and the role to which agents targeting there, like PARPs or others, will be able to synergize with IO. I'm going to hope that that also begins to broaden to other types of mechanisms as more and more is understood about what underlies the generation of those mutational loads or lack thereof. At least on the tumor side, there's obviously still the whole tumor microenvironment aspect that has to be addressed. And I'll also just add to that, though, is kind of the flip side. Just playing somewhat devil's advocate is, just as we mentioned earlier, kind of the evolving role or the rapid sea change that happened with the BRAF inhibitors versus the checkpoint inhibitors, say in melanoma. There are many studies that have begun to show that when you start to combine some of these IO modalities, you may I underscore, start to obviate the need for some of these stratifications, whether those are more IO-directed stratifications like PDL, because certainly there are studies showing combinations of checkpoints and co-stims or others can become independent of that PDL status. And that may even ultimately apply to some of these other mechanisms, just like we've seen with BRAF inhibitors. So it's going to be a very interesting time and very dynamic, which is part of why we're having this discussion.
Yep. I just wanted to add to this, just since we're on prostate cancer, is that with a tumor like that, and you can also talk about breast cancer in the same sort of respect, and maybe even ovarian cancer, but with prostate cancer being such androgen-driven cancer, I'm not sure if we're done with the different ways to inhibit androgen-driven progression of the disease. I think there's still some other interesting agents out there that may be a non-IO story. They're coming in sort of like the alternate splice forms of the androgen antagonists, and what came out of Marion Sadar's lab, and I think it's now in ESSA Therapeutics. Looks like another sort of interesting another strategy to come at it from the endocrine side of things. So.
Yeah, and certainly with Tokai and their triple blockade of the androgen receptor pathway.
Right. Yep.
Yeah. Okay. So the final theme here, and 2 slides left, so hopefully you'll stick with us to just past the top of the hour. Drug and the undruggable. So the first example of this is miRNA, and I'll let Mike speak to the specifics of this. Certainly, this is a platform that has been it's been in the R&D stages for a while. I think delivery has been kind of an open question, as well as survival once you kind of get into the serum and the in situ situation. But this promise of being able to go after some of these classic oncogenes, or in this case, an miRNA pathway that is upstream of some of these different pathways, some of these different molecules. It's something that people haven't given up on, but it's also been somewhat overshadowed, I think, if you can believe that or not. Targeting MYC, targeting P53, it's kind of been the dream forever. Do people still care about that?
Yeah. So certainly. So I'll speak a couple things about this. So one is that Joel's mentioned one of the big limitations in using RNA as a therapeutic, or oligonucleotides, or any sense, or whatever approach, has been challenging from a delivery standpoint, and it's really driven a lot of companies to exit the field. Pharma has gotten out and back in, and out and in at times, but really kind of deprioritized a lot of the things in the RNAi and antisense space in oncology. I think things are changing now, and so a lot of that is driven by the new promise of RNA as a therapeutic. If we look at the war chest that Moderna has amassed in enabling therapeutics across therapeutic areas, and oncology may be one of the last areas that they get into in their spin out on Onkedo that's focusing. It's a little bit unknown exactly what the strategy is going to be in oncology, but certainly there's significant investment going into enabling RNA therapeutics. Now, on the other side is the microRNA. So microRNAs is really emerging, and it's not as established science as knocking down messages or going after the MYC oncogene that was the first oncogene described in humans and has been elusive all these years. So there's great interest in RNAi as being able to make that a druggable target after all these years. So what Dicerna is doing and whatnot looks like a very interesting program just by its uniqueness and the role of MYC in multiple tumor types. But so if we go back to microRNAs as has emerged as a prognostic tool for looking at predictive markers for progression. It's been looked at for monitoring disease, response to therapies and ways to stratify patients. But certainly, it can be used to also get at those sort of undruggable targets. And so, what's emerging from Myrna looks like a very interesting program. It's the first microRNA into clinical trials, and so that in itself, I think, is a very unique thing. And so I've always looked at it as being very distinct. If you put aside the toll, the toll story around the oligonucleotides driving immune response, it's really been largely a non-immune story. However, you look down at this, bottom bullet point, talking about their financing that they raised to move into phase 2 trials with the program. Again, here's an immuno-oncology story that is now emerging. They publication recently showing that inhibiting miR-34 regulates PD-L1 expression. So again, we have another sort of interesting synergy with immunology. So
yeah, Jeff, I'm just going to move to the last slide, and I'll let you speak to it. I mean, we've had this kind of rash of raises and deals associated with these platforms going after that promise to at long last address some of these undruggable targets, the mix, the transcription factors of the world. Whether it's warp drive being fostered by Sanofi Sunrise, and Sanofi recently announced that they were kind of revising the terms of that relationship, but nonetheless, remain very interested, particularly in their SMART platform and their RAS inhibitors that are coming out of that. Arvinas, with technology coming out of Craig Crew's lab, kind of leveraging the proteasome degradation machinery, and T4 similarly with a platform that likewise looks to or promises to attach tags and degrade different oncodrivers, oncogenes, etc. With the promise of being able to address some of these undruggable targets. On the face of it, these are not IO stories. They're kind of standing out, at least in my mind, as kind of going back to the original promise of targeted therapy, but certainly being recast in a new light. Do you agree with that, Jeff, or is this ultimately all going to kind of blend together with what's happening in IO?
Well, I mean, I think I think on the surface, absolutely. I mean, I think it's hard to speculate, from what might ultimately happen in terms of any scientific crossover. However, it is tempting to speculate that beyond the application of these platforms to these classic undruggable targets, which are being approached by other modalities like RNA, so that would include MYC. And RAS and P53, although there are some current programs kind of targeting those that aren't oligo-based. It kind of raises in my mind whether 2 points. One goes back to your earlier slide talking about the distinction between IO and non-IO and what they do, and it underscores the important distinction between what IO can do versus what, at least for the moment, we think it cannot do, and why you need additional modalities with better therapeutic windows to come in early to gain control of aggressive disease, or to use as part of these kind of upfront attempts, because insofar as we are still kind of stuck in identifying most cancers when they are advanced stage, we're going to need to use surgery, radiation, and some of these targeted agents to gain the control to allow the time for many of these immune agents to act. The other aspect is much more speculative, which is just like we've seen a lot of interest around CRISPR and gene editing, and we've all talked collectively, and there's been some talk in the industry of kind of if you really got really sci-fi and went out there, could you envision a world where you could go in and do in vivo in the person or in the person's cancer. Some of these modulations that we're doing now with adoptive cell therapy or checkpoint inhibitors, et cetera, could you kind of go in and do some of those modifications with CRISPR? I think also there's the idea that, if we have some additional new modalities to target these undruggable targets, those also might be able to be applied to this idea of targeting additional genes and pathways that are involved in both the tumor side of co-opting and the immune system, as well as the tumor microenvironment, et cetera, but not at the DNA level like CRISPR, or not even at the not necessarily at the RNA level, although clearly kind of modifying transcriptional regulators would do that as well.
Great. All right, so I think this kind of brings us to the end of the presentation, or at least the slides. Maybe it's appropriate to end with a discussion about a quick discussion about white space. Are there areas, and I'll start with Mike on this. Are there any areas, either from a therapeutic area standpoint, a setting standpoint, or perhaps a combinatorial versus go-it-alone standpoint, where there is white space and one could potentially position themselves for success in this rapidly changing landscape?
Well, two-part question. So one is the white space, and the other is how do you position for success? Since I spend more time in heme malignancies, I can throw out some areas that have been challenging, or at least there hasn't been much progress in immuno-oncology, and the space seems to be dominated by chemotherapy still and pathway inhibitors, and Jeff can comment on some other things. But certainly, probably of all the heme malignancies and white spaces, it's been, AML has been a, unsolvable story for many years, and there's relatively little activity in immuno-oncology, even in that setting. Heme malignancies have been really impacted, B-cell malignancies, by the T-cell therapies, both CAR-Ts and some interesting, early data and TCRs against, other malignancies look quite interesting. But still, I mean, there's really not a lot going on in the AML space. I think one of the things unique about CD19 is that, you can live without B cells, but you can't necessarily live with many of the targets that you would develop an adoptive T-cell therapy against for AML. So there's been very little activity. I think CureTech has a combination therapy with an HDAC inhibitor, azacitidine, that we'll follow and see how. I think it's early yet, but it looks like there is some attempt at something. There's some CD1. I think Cellectis has a CD123 UCART program. Again, it's not in the clinic yet, as far as I know, so it's very early. Yeah. But so as yet, the programs are really driven by the molecular drivers that have been identified through the whole genome sequencing of AML patients. It's one of the most molecularly characterized disease, even though it is a very heterogeneous patient population. But we're following the different targeted epigenetic agents that look interesting, and they don't really, I mean, you can probably build an IO story around them, but that's not really the way that they had entered the clinic. That they were really, IDH1, IDH2, the domain inhibitors, BET inhibitors, yeah. DOT1L, they all came in there based on this very compelling genetic evidence that they were molecular drivers of the disease.
Yeah, and I think the prevailing dogma is that AML, like myeloma, is regarded as heterogeneous enough disease that you kind of have to figure out where that balance is between targeted therapy, where the disease is going to find a way around it, and chemotherapy, where you can achieve perhaps responses. Durability is an open question, but safety, of course, is an important one. We did have on that note, we had a question from one of our audience members about has chemotherapy been successfully combined with IO Jeff, do you have any analogs that, or any examples that stick out in your mind?
Well, I mean, there's a lot of studies ongoing, but certainly you can point to cyclophosphamide on one hand in kind of the heme space. I already referred to gemcitabine. There's actually some data around the role of taxanes in the space. So yeah, there certainly are examples, and I think part of it is also, and there are new companies that now are trying to, there's obviously a long history of this idea of drug delivery or reformulation around cytotoxics, and frankly, a lot of failures trying to demonstrate any real significant benefit, except for a few, outstanding, exceptions like a Abraxane. But there are kind of a new wave of companies exploring cytotoxics and chemotherapeutics and either modifications to those or ways to deliver those, et cetera, that emphasize, that come from the mindset of immunotherapy, the idea of trying to get that immunogenic cell death, trying to maximize that cell killing that's going to lead to infiltration most likely in the context of checkpoint inhibitors, while at the same time kind of minimizing a lot of the side effects associated with chemo agents. So, as I said, it's an active area that when we've done our analytics around where all these combination studies are going, it's still a sizable chunk are looking at combining IO agents in some way with chemo. I think the challenge there, and it's not just in combination with chemo, it's any type of IO combination, or frankly any type of combination in oncology is, historically we've done too little exploration for a variety of reasons, mostly logistical, in terms of how best to combine, when and how to sequence, and the timing, et cetera. And there certainly are, even just in the case of IO, very clear examples of where you get better synergy with certain sequencing of those agents, and conceivably even antagonism, and frankly there are examples of that with cytotoxics too.
And certainly, short course, like you pointed to, cyclophosphamide is thought to be essential for enabling, some of the immunotherapy approaches by getting rid of Treg cells and myeloid-derived suppressor cells, so that you can enable whatever immunotherapy modality that you have to kind of, take the brakes off before, you put in your adoptive T-cell therapy or checkpoint inhibitors or whatever.
Yeah, and I want to go back to also to your question that you started with to Mike about where the white spaces are. And I think there's a lot of white spaces still. I mean, there are still large segments of patients in the quote unquote more immunoresponsive tumors who outside of maybe of the Ipi-Nivo combination in melanoma, who could benefit either from raising the percentage of patients who have durable remissions, as well as the idea of doing so with an improved therapeutic window around those combinations, and that extends clearly also into the less immunoresponsive, or the quote-unquote cold tumor types. So there's lots of unmet need remaining, and from an even more fundamental standpoint, something that has kind of been referred to but we haven't dealt on directly is that the heterogeneity that is a defining aspect of tumors is something very critical here, that calls for not only the ongoing need to have, say, multiple more directly cytotoxic agents or cytolytic agents or combinations of targeted agents to use, with in advance of or with IOs ultimately, but being able to deal with that heterogeneity. I mean, there was a recent in the fall article in PNAS talking about the amazing genetic diversity in HCC, for example, and I think this is something that will still need to be addressed. The sheer amount of diversity will require an ongoing, and that's part of the attraction, I think, and part of the value proposition to the immunotherapeutic approaches. The idea being that the immune system is as plastic and malleable and adaptable as the tumor itself is.
Great. All right, well, we are at the quarter of the hour. I want to really thank everyone for sticking with us. Hopefully you're able to take something useful away. I want to thank my colleagues, Mike Rice and Jeff Bachman, for this very interesting, provocative discussion. And, you know, we really hope to see some of you or all of you in New York City at Cancer Progress in a couple of weeks. There's still a few tickets left. If you haven't gone on the website, please go on, take a look. We have a great program lined up, 2 days. It's going to cover the, you know, the spectrum of clinical, commercial, scientific, and it's really going to be incredible. So again, you know, we really thank everyone, and have a great rest of your weekend. Thank you so much.
Yep. Thank you, Joel, for great moderating, and thanks everyone for staying tuned to this discussion.